Welcome to the WTR Small Cap Spotlight podcast. I'm your host, Tim Gerdeman, Vice Chair and Co-founder and Chief Marketing Officer of Water Tower Research. In today's podcast episode, I'm being joined by Dr. Maria Maccecchini, CEO of Annovis Bio, NYSE ticker symbol ANVS. Annovis Bio is a late clinical stage drug platform company developing novel disease-modifying therapies for neurodegenerative diseases, to include targeting Alzheimer's and Parkinson's, as well as other related diseases. Also joining us today is my Water Tower Research Equity Research colleague, Robert Sassoon. Good morning, Maria and Robert, and thank you for joining today's podcast.
Thank you so much for having me. I'm so happy to be here. Thank you.
Thanks, Tim. I'm really looking forward towards this conversation.
Likewise. So, Maria, before I turn things over to Robert to ask several questions, let me kick off the podcast by asking you to tell our podcast listeners about your professional background and how your interest in neurodegenerative diseases evolved.
So I'm a scientist, and when I was 23, I was looking at cell death and at cell growth. I was always really interested in science, and I realized that nerve cells don't die, whereas all other cells die. And also, once nerve cells die, that's it. Whereas other cells, they can regenerate. They die faster, but they also divide. And so I decided I was going to actually work on nerve cells and figure out how to keep them from dying, because once nerve cells die, that's it. And I was 23. And then I was looking into how to study that and figured out that there was no way I could study it because really we didn't understand anything. And also in those days, it was impossible to keep nerve cells in culture. You couldn't really study them. You had to open the brain. That's the only way you could look at them. So I became a biochemist, and then I became a molecular biologist because that was the hot thing to do, genes. And I went into neuroscience, and I've been in dying nerve cells forever. In fact, my first company that I started in 92 was supposed to protect nerve cells from dying. It didn't quite work, but in fact, this company now protects nerve cells from dying. So I'm getting there.
I have to give you accolades for that intellectual curiosity at such a young age, but that having been said, I'll turn it over to Robert.
Hi, Maria. I think you're really operating in a fascinating area, and I think we're just now beginning to see some really innovative breakthroughs that are giving some hope to many millions of suffering from neurological diseases, which were previously, just a pipe dream for them. So, kudos to you. So, before we actually get into your clinical programs, as a lead-in, can you summarize the scientific basis of Buntanetap, your lead candidate, how it works, and what you think differentiates it from other disease-modifying therapies that are now available for Alzheimer's and, plenty of others that now in the development pipelines for Alzheimer's and Parkinson's. Those are the sort of target areas that you're specifically focusing on.
Yes, going back to nerve cells that die. How do they get killed? Now, of course, you can hit them over the head and they'll die. But as we all know, and we have known now for 30 years, A beta or plaques kills nerve cells. And in fact, the two disease-modifying drugs on the market today do attack plaque. Now, while A beta and plaque kills nerve cells, tangles, which is tau, alpha-synuclein, which is Lewy bodies, a number of proteins that make blobs, that make these aggregates, they kill nerve cells. So just attacking one of these aggregates is not good enough. And in fact, if we look at the two drugs that are on the market, they improve cognition 30% over placebo. But that is a little misleading because 30% over placebo means that placebo goes downhill 100%, and 30% better is minus 70%. So actually, these drugs slow the decay of the cognition, but they don't improve it. And if you think that there are a number of toxic proteins in the brain, just attacking one is not good enough. And that's exactly what we see. Our drug attacks plaques, tangles, Lewy bodies, and other aggregates.
Right. So back in April, just carrying on from that, back in April of 2024, Annovis released data from a phase 2/3 study of Buntanetap in Alzheimer's. So, involving subgroups of patients and at different stages of disease progression, which created some confusion and maybe you could say disappointment, leading to substantially negative reaction from investors reflected in quite a sharp drop in the share price. However, the program has since moved on and been given the green light by the FDA to proceed to phase 3. I believe that you've already started dosing some patients in that program. So I have a few questions, and 3 questions here. For context, can you highlight briefly the key concerns from the results that elicited that reaction, and to what extent this was based on misinformation or misinterpretation, at least, of the data? Secondly, are there any lessons that you've taken from that episode in coming up with appropriate clinical trial design, protocols, and primary outcome targets of the phase 3 program? And thirdly, given the planned enrollment size of, I believe it's 750 people for this program, can you give us an indication of top-line readout and NDA submission timeline targets for the program?
Yes, absolutely. So the issue with the phase 2/3 Alzheimer's study was that it did not work in everybody. When you do a study, you tell the FDA, this is what we're looking for, and the FDA expects you that you show this effect in everybody you recruit. And that is what we were not able to show. Number one, we had patients in our study that did not have Alzheimer's disease. We should have done a better job predetermining the patients. We relied on the doctors to say whether patients did or did not have Alzheimer's disease, and some doctors just took patients in whether they did or didn't have Alzheimer's disease. So the study, as I said, in the whole population didn't work. However, when we then specifically looked at early Alzheimer patients, then we saw beautiful dose-dependent statistical significant data. And the reason the FDA gave us the go-ahead is that our new study is in this group of patients, the very group where we have highly statistical significant data. Now, how did we get from the total population to this group, and why did the FDA agree with it? While we were doing the study, a new test was invented. And the new test can determine from your blood whether you have Alzheimer's or not. As you know, 10 years ago, we would say the only way to really diagnose Alzheimer's is to open your brain after you're dead. And then we started doing PET scans, because PET scans image plaque in the brain. And that's actually how the 2 approved Alzheimer drugs got on the market. We did not do PET scans. However, as I said, this new plasma biomarker, which is a P217 tau biomarker, was invented and approved by the FDA. So after the fact, we were able to look at all our patients and decide which ones had Alzheimer's and which ones did not. And the patients that have this really nice response to our drug were selected with this p-tau assay. So when we went to the FDA and we told them, if we take the group that has the p-tau assay correct inclusion, we see this data, they said, go ahead, this is fine. I'm not sure the market really understood the p-tau assay because it's a brand new assay. We are the first ones to use it in a phase 3 clinical trial.
Right.
And then you wanted to know, your third question was, where are we with the study and when will we be where? So we have recruited almost 15 patients. We have opened 50 sites. We still have, we say 100, but 80 is good enough. So we still have another 30 sites to go. And we expect the readout for the 6-month study to be in the summer of 26, and the readout for the 18-month study a year later, summer of 27.
Okay, great. Just to follow up, Biogen and Eli Lilly are actually doing pre-symptomatic trials. They conduct pre-symptomatic trials, phase 3 trials for their approved drugs. Is that something that you going to think about doing in parallel with your current trials?
I actually would love to do a prevention trial because we have beautiful data. Unfortunately, in a short experimental endpoint, we weren't expecting to see that in Parkinson's. In the whole Parkinson's population, every single last Parkinson's patients, I know I'm jumping to Parkinson's, we saw that cognition deteriorates a little bit in the whole Parkinson's population because people get old, their cognition goes down. In the 2 treated groups, cognition remains identical. That really shows prevention. If it was an 18-month study, I would say it's a preventative. So
Right.
yes, we'd like to do that, but after we have efficacy.
Certainly in Alzheimer's, I think it's a huge market because I've seen estimates that, at the moment, estimates of diagnosed Alzheimer's is around maybe 40, 50 million worldwide. But I've seen an estimate that that they're pre-symptomatic, that could start, 20 years earlier. Is the market could be as high as big as over 300 million people. So that's a huge market that will be
Yes,
big.
and I think our drug also lends itself very well for prevention because it has no side effects. It's very, very safe.
Great.
It really is. We have treated 1,000 patients and it's very, very safe. And number 2, it's a pill a day, which makes it easy.
Great. Yeah. You mentioned Parkinson's. So we also had some phase 3 results from your Parkinson's program. You released those, I think, sometime in the summer of last year. And they demonstrated that in patients that specifically had a cognitive deficit, cognition improved. And that in that specific patient population, the original primary and secondary endpoints as per MDS UPDRS, and I'll just break here that that's a scale that's widely used in Parkinson's to measure motor and non-motor symptoms. But you met all the primary and endpoints and the secondary endpoints in that for that particular population. So what are the next steps for that particular program?
So you didn't mention the negative part of the Parkinson's study. Just
I'll
like in
leave that
the
to you.
Alzheimer's, we had non-Parkinson's patients in the Parkinson's study. So again, the whole ITT, everybody did not work in the UPDRS. However, as I just mentioned before, in the whole ITT population, the whole 530 patients, cognition worked. Cognition worked in Parkinson's in the whole population as a preventative, and in the ones that were cognitively impaired as an improvement. So from that point of view, we feel very, very strongly that the drug works in cognition, period. It doesn't matter in what population. Now in Parkinson's, we have looked at the other Parkinson's population where we have non-Parkinson's patients and we have a per protocol population. We eliminated the patients that did not follow protocol. And in those patients, we did meet UPDRS 1, 2, 3, 4, and total. So it worked in a slightly smaller population. Unfortunately, just like in Alzheimer's, that's a subgroup.
Right.
And so we are going to the FDA, but first we want to go to the FDA. In fact, we are just sending them a letter for cognition because Parkinson's has a lot of cognitive problems in subgroups. There are groups of Parkinson's that are called dementia with Lewy bodies. The groups of Parkinson's are called Parkinson's disease with dementia. And these groups actually have Alzheimer component to their Parkinson's. So it's not clean Parkinson's, it's a mix of Alzheimer's and Parkinson's. And remember, I was saying that the brain is full of a number of toxic proteins, not just A-beta, not just tau, not just alpha-synuclein. And in these groups, they have everything. They have every aggregating protein present in Alzheimer's, present in Parkinson's at high levels. because in Alzheimer's, there are Parkinson's proteins. In Parkinson's, there are Alzheimer's proteins, but they're lower level. Whereas in Lewy body dementia, they're all at high levels. So these are good populations for us to look at cognition.
Right.
That's the first step. Now, we still want to go back and do a Parkinson's study in the regular Parkinson's population. And there, we're going to have to measure alpha-synuclein in spinal fluid.
Right. Yeah, I guess because unlike Alzheimer's, the biomarker market in Parkinson's is not very developed. So just as a follow-up on that, you mentioned the connection with Alzheimer's and the Parkinson's trial. Is there any? I mean, can you actually use any of the data that you've got out of the Parkinson's trial for your phase 3 Alzheimer's program?
No, but we're using the data from Alzheimer's for our not yet designed Parkinson's program.
Okay, right.
Because in Alzheimer's, we have tested a lot more biomarkers. We have really analyzed our data very, very well. And in fact, right now, we have gone back to our Parkinson's study, and we are looking at all the biomarkers that worked in Alzheimer's. We are looking at them in Parkinson's also. Okay,
great. So let's move on to the. I'll have one more question or questions related to the financing, because that's really the lifeblood of any biotech company. So last month you raised $21 million in a public offering. So where does this actually take you with respect to your goals of advancing your lead programs, and how quickly will you need to supplement your funding?
So it's supposed to last until we have the 6-month study done summer of next year. If, in fact, we don't do anything else, which right now we tabled the Parkinson's, we tabled the open label, we're waiting for have more money. So we'd have to raise money middle of next year.
Right, okay. But you have various options, I think you've mentioned in the recent investor broadcast. So you're talking about $200 million of...
Well, if I had 200 million, I would not be where I am. Okay. No, that did not materialize. But we have some more options, yes, but they are not very imminent, yes.
So it's a wait and see.
Yes.
Okay, well, I think I'll hand over back to Tim if you've got any other questions you want to ask.
All set. That was actually a really fascinating discussion. So thank you for joining us today, Dr. Maria Maccecchini, CEO of Annovis Bio, NYSE, ticker symbol ANVS. Also, thanks to my equity research colleague, Robert Sassoon, for joining us today. And finally, a special thanks to the producer and editor of the podcast, my colleague, Joe Brunetto. Thank you for listening, and don't forget to subscribe, as well as visiting www.watertowerresearch.com to stay up to speed on the company's small cap written research reports, podcasts, fireside chats, and conference schedules. We'll see you next time for another edition of the WTR Small Cap Spotlight podcast.
Very much.